Jeff, it's all yours.
Well, thank you for that very kind introduction. So I'll be talking about those three groups, but in the order of humans and then birds and baboons. So you can choose to tune out if one or more of those are of less interest to you. So I think I'll start with sort of the human side and what originally motivated me about Admixture. And I'll start by sort of describing sort of a cartoon version of what we think about human evolution, which is not actually that much. So it's thought that our ancestors, or what we would first think of as sort of humans, originated probably somewhere in East Africa 2 or 2.5 million years ago, and those individuals then spread probably throughout the rest of Africa into tropical Eurasia, and then into parts of temperate Eurasia. And then there's sort of a gap where we don't know too much, but there's sort of another wave that happened where we think that modern humans, and there's some difference of opinion of exactly what that means, that they evolved somewhere either in East Africa or Southern Africa, but much more recently, and they also spread from throughout Africa into Eurasia and Australia, finally up into Siberia, across the Bering Land Bridge at that time, and down into the Americas. And so the question that I've been interested in since a graduate student is essentially what happened to the first wave of humans that left Africa. So they then populated Africa and Eurasia, and were there when modern humans came through at a much later point. And this has been sort of a subject of debate for a very long time, and sort of the axis over which people have thought about this is the extent to which modern humans interbred with the indigenous hominins that were there, or what we usually call archaic human groups. Did they interbreed? Did modern humans wipe them out? Or what combination of those two happened? And since I usually think of things in genetic terms, so you can think of it as if we look at our genomes and trace back our history from our parents, grandparents, great-grandparents, and so on back to 50,000, 60,000 years ago, are our ancestors modern humans exclusively, or are some of them also these archaic human groups, such as Neanderthals, for example? And so there are sort of two different ways that you can approach this. You can, if you're lucky enough to have a genome sequence from an extinct archaic human group, then you can use that directly to sort of identify how much, if any, admixture there had been in the past. But I'm not going to talk about that really. I'm going to talk more about an indirect approach that people use, including myself, that looks at patterns of genetic variation and tries to analyze that to look for evidence that admixture might have occurred. Okay, so just as background, sort of the direct approach has suggested there's been at least two major sort of interbreeding events that happened in human history. One where, so Neanderthals here must have interbred with so Neanderthal DNA is found in all non-African groups, and the sort of most parsimonious explanation for that is that there's some interbreeding between Neanderthals and the ancestors of non-African populations. And then there's also a separate archaic human group called Denisovans, which are mostly known from a single fossil found in a cave in southern Siberia. And it turns out, for some weird reason, that their DNA shows up in modern-day Melanesians, but essentially nowhere else. And so that sort of could be explained by some separate admixture event between Denisovans and the ancestors of Melanesians. Okay. So the indirect approach that I'm going to talk about, the basic idea is something that's often called admixture mapping. So you can think of it as if we were to show up in Europe or parts of the Middle East like 40 or 45,000 years ago when modern humans and Neanderthals were contemporaneous, if we could sample those individuals, their genomes would be very distinct from each other. And we could tell them apart very easily from DNA. And so if you imagine what would happen if they were bred, here would be an F1, they'd have one homologous chromosome inherited from a human parent and one from a Neanderthal parent. And as time went on, an F2 would look similar, except then the genome of an F2 would be a mosaic of stretches of chromosomes that were inherited from human grandparents versus Neanderthal grandparents. And generation by generation, you might expect that the blocks of ancestry would get smaller and smaller. So there's recombination happening each generation, and that process of recombination breaks up each chromosome into smaller and smaller blocks of ancestry that could be traced back to either modern human ancestors or, in this case, Neanderthal ancestors. So there's been a lot of generations since that admixture might have occurred. And so that means that if you're looking for stretches of Neanderthal ancestry, these little red bits, by now they're pretty small, on the order of tens of kilobases long. So when you compare that to a genome that's 3 billion base pairs, it's very small. But on the other hand, you can think of it in terms of a 50 kilobase chunk is actually a substantial amount of sequence that you might hope to identify purely by looking at modern human sequences. So I'm not going to talk too much about ways that you do that, but I will sort of describe some of the findings that we've had. So we took some whole genome sequences that are publicly available from a bunch of different human self-identified ethnic groups, and the basic idea is that if you find a fragment of a genome that actually is red, it's going to be quite different from other homologous sequences from other individuals. And that would show up in terms of what are called patterns of linkage disequilibrium. I mean, roughly speaking, if I knew that I had Neanderthal DNA at a certain stretch of my genome, that Neanderthal DNA is going to look pretty different from everyone else's modern human DNA at the same region. And so you can tell that by looking either at sort of patterns of diversity of my genome versus other people's, or patterns of linkage disequilibrium of the alleles that would have been inherited from Neanderthals compared with the other alleles that are in the modern human population. So I don't want to talk about that too much, but there are sort of ways that you can do this. And you can essentially comb through the genome and find regions that look very unusual, that are outliers in some sense. And you can show that essentially those types of regions, the most extreme of which are extremely unlikely to have arisen by anything else except for something like ancient admixture. Okay, and if you sort of combine that information that comes only from modern human sequences, you can also then use the Neanderthal sequence that we have. So if I have a hypothesis that a certain stretch of DNA actually was inherited from Neanderthals, well then you would expect to see that sequence in the actual Neanderthal genome. And it turns out that you would also expect that the Neanderthal alleles would be mostly derived, which means different from the ancestral type or chimpanzee, for example. And that's sort of to distinguish recent admixture from some kind of incomplete lineage sorting. And then the last thing that we would know is that if you think that a certain region or certain alleles are inherited from Neanderthals, you would expect to find them primarily in individuals of non-African descent, and for those alleles to be rare absent in sub-Saharan African populations. So we sort of had a process by which we identified regions that we thought were inherited from Neanderthals, and it turns out that the number of these regions that we could identify differed depending on what group you sampled. So here are the different groups. These are Biaka Pygmies, Niger-Congo speakers, including mostly Yoruba, I think. These are Maasai, individuals of European descent, East Asian descent, and South Asian descent. And the only thing that I want to sort of point out as well, there actually seems to be a difference between the amount of Neanderthal ancestry found in Maasai versus other African groups. And that it turns out that there's a difference in the amount of Neanderthal ancestry in European individuals versus Asian ones. So, and it goes in the direction that was not obvious to people beforehand, namely that East Asian individuals generally have more Neanderthal ancestry than individuals of European descent. So, I have more Neanderthal DNA than most of you, is the basic summary. And that was not obvious to people because we knew of Neanderthals primarily from fossils found in the Middle East and in Europe, and a little bit in West Asia. And there are no known Neanderthal fossils from East Asia. So the simple view was that modern humans and Neanderthals were meeting and possibly admixing in Europe. And now, because of this and other findings, the current belief is more that modern humans and Neanderthals interbred probably in the Middle East, sort of during the time period when modern humans first left Africa. And somehow, during that process, the ancestors of the Asian populations over there were either in contact with Neanderthals more or interbred with them more than the ancestors of Europeans. Okay, so this is sort of an interesting fact, but I guess the thing that I want to talk about today is that the slide I put up about sort of admixture and understanding how it works is one that's a lot more general than just looking at human versus Neanderthal ancestry. So for example, you could look at other human groups. So African Americans, you could think of them as individuals that have some ancestry from sub-Saharan African groups and some ancestry from European groups. Latinos, same thing, ancestry from potentially several different groups, and other species too. So it's the same thing whether you're looking at humans or you're looking at chimpanzees or you're looking at baboons or you're looking at just about any species you can think of. It's not uncommon that you have populations that are genetically distinct from each other that at some point come into contact and produce, F1, F2, and other admixed individuals. And whether you call them separate species or subspecies or populations, it doesn't really matter. The point is just that from a genetic standpoint, it's not uncommon to have distinct genetic groups come into contact and interbreed with each other. So that's what I want to talk about, a couple other examples of that. So first, in humans, back to the Maasai, who I showed have some Neanderthal ancestry, there's an explanation for that. An explanation, so Maasai they're pastoralists, they're native to East Africa, and there's been some suggestion before that actually when you look at their genomes, they have a substantial amount of non-African ancestry. So what you can do is estimate across the genomes of the Maasai their local ancestry of what parts of it might have sort of an African component versus a non-African component. And I guess I'm a little loose with words. When I say African, I usually actually mean sub-Saharan African, and non-African would in this case include North Africans.
Jeff, is that recent or ancient?
It is on the scale of 2,000 or 3,000 years ago, so intermediate. So the basic process is this. So you could take a Maasai genome, and so you can think that their actual ancestry might be some mosaic of African and non-African components. And so if this were actually what an individual's ancestry was, our goal would be to infer across a chromosome or their whole genome how many of their two alleles would have African ancestry. So you can see here it's 1, and then for this stretch here it's 2, where you have 2 black, homologs there in this case. Now, it's a bad choice of color, I do admit. Then down to 1, up to 2, 1, 2, 1, and 0 as the ancestry shifts across a chromosome. And what you can do after doing this kind of inference is you can take an admixed individual's genome and partition it into regions of the genome that might have either 0, 1, or 2 inferred African alleles. Okay, so this is what we can do in practice. We can start with genetic data. We don't actually observe this true history, but we can infer it pretty accurately, and then we can use that inference to partition their genome into different parts. And it turns out that when you look at those different parts, they actually look pretty different. And so this is measuring. So there's a measure of sort of Neanderthal ancestry that was developed by the original Neanderthal Genome Consortium. That's this D-statistic. And the only simple way of thinking about it is the more negative it is, the more Neanderthal ancestry you have. Okay, and so what we find here is if you take the regions of the genome that we think have 2 non-African alleles, they have very negative d-values, and which is consistent with their having Neanderthal ancestry. The regions of the genome that have 1 African allele and 1 non-African allele are pretty much completely intermediate, and then the regions of the genome that we think have 2 African alleles, they have no evidence of Neanderthal ancestry. So this is sort of exactly in line with what we think, and the sort of hypothesis that's been thrown out there is that there's been a substantial amount of trade and contact and potentially interbreeding between Arab or Middle Eastern traders along the coast of East Africa, all the way down to Southern Africa, in fact, over at least the past 2,000 years. So this admixture, when you if you look at sort of how long these segments are in terms of genetic distance, you can use that to estimate how old you think the admixture must have been. So roughly speaking, the older it's been, the shorter these fragments are. And if you do a very rough back of the envelope calculation of when that admixture must have been, it's on the order of 80 to 100 generations ago. So a couple thousand years ago, roughly speaking. So let me give another example of sort of using admixture to look at something that may be interesting. So if you take, so in humans there's this large project called the HapMap that that happened 5 or 10 years ago where they took individuals and they generated large-scale genotype data from them. And some of the samples consisted of trios from Mexican-American individuals sampled in Southern California. And if you look at the genetic ancestry of individuals that are Mexican-American, it's primarily some component that's Native American ancestry and some component that's European ancestry, to a first approximation. And it turns out that if you look at trios, so you're sampling parents here, and you look at how much Native American versus European ancestry they have, there's a correlation. So this is an old slide, but this sort of shows up again and again when you look at more data, that the amount of Native American ancestry in a mother correlates with the amount of Native American ancestry in a father. So you can think of this as some degree of assorted mating, like genetic type. Now there are many possible explanations for this, so I'm not going to go into that. But this is a pattern that's there. It's a pattern that shows up in African American trios as well. Okay, so I'm going to switch gears a little bit. I'm going to switch gears because humans are unusual in many ways. And as a scientist, the main way in which they're unusual is that there's a tremendous amount of money. So, NIH, and they support a lot of research. So doing the kind of analysis for looking for Neanderthal ancestry, for example, is something that you could not easily do in other species. So part of my interest has been, you know, we have some methods that seem to work well in humans, but how do they work when you have some other species? So I'm going to talk a little bit about some work that we've been doing. This is work looking at potential admixture between spotted and barred owls with Zach Hanna, who should be somewhere in the audience here in the very back. And Jack Dumbacher, who's at the California Academy of Sciences. Okay, so you probably already know this is a picture of a spotted owl. They're found throughout sort of the western North America. Up here would be the northern spotted owl, which is listed as an endangered species. So they're in the news a lot, and there are several reasons why they're endangered. One is that their habitat is mostly old-growth forest, which is rapidly disappearing. And another potential reason is that there's now, within the past 100 years, an expansion of barred owls that are native to the eastern side of North America. They have expanded across here, and now their range almost completely intersects with the range of the northern spotted owl. And this is significant, one, because the range expansion is recent. As far as we know, it's pretty recent. But also that barred owls tend to outcompete spotted owls whenever they're found in the same location. Okay, so that's what we know. What we don't know is sort of whether barred owls and spotted owls interbreed with each other. And so here is a spotted owl. This is a barred owl. So you can look at, so ask Zach, because I actually don't know this very well, but if you look at their feathers, the sort of light areas here kind of look like spots. You look here, it kind of looks a little different. There's much more white and just long streaks of dark. And this in the middle is a barred owl from Siskiyou. And in many ways that owl looks to be somewhat intermediate in terms of the coloration here, in terms of overall body size, intermediate. And because of that, it's a hypothesis that this individual might actually have ancestry from both spotted and barred owls. And so this is something that we wanted to be able to test. Because it would, it sort of has evolutionary ramifications and also conservation ones. Okay, so here are some of the challenges, which come down essentially to the last one. Money is limited. So there's actually no genome available. Other genomic resources are actually pretty poor in terms of a physical map, a genetic map, etc. The work that I talked about earlier usually requires that you know the haplotypic phase of sequences. Here, you're generally not going to know that. And all of this comes down to, if you had a lot of money and you spent $10 billion gathering sequence data like you've done in humans, well, we'd know a lot. We'd have a very good genome. We'd have great genomic resources, and this question would be very easy to answer. So essentially the question comes down to, well, if you have a certain amount of money and you want to address a question like admixture, how do you do it? I don't know if I'm going to have a great answer for that. What I'm going to start with is that you kind of need to have some genome assembly. And here the problem is that we can generate lots of cheap sequence data using like Illumina, but that Illumina sequence data comes in 100 base pair chunks, and a genome is much larger. And in the case of spotted owl, it's actually on the small side, which is only about 1.5 billion base pairs. Small at least for the range of vertebrates. Okay, so one of the issues surrounding that is when your reads are very small, and so the standard way that people sequence is something that's called shotgun sequencing. So it's essentially you take genomic DNA and you fragment it randomly, and you sequence these random bits. And what that means is you're not targeting a specific region, you're just targeting everything at the same time. And when you have small reads, so this is sort of a little cartoon that I'm not sure it works so well, but when you have small reads, the problem is that even when you sample random locations, by chance you'll get gaps where you don't sequence something. And so in practice, when your reads are very small, you're still going to have gaps. If your reads are very long, then with the same amount of coverage, you're much more likely to cover everything. Okay, so this is just to say, okay, well here we have sort of an average of 3 times coverage here, an average of 3 times coverage there, and here where the reads are long, you've covered everything. Actually, you've covered everything twice. Here, the reads are long. Sometimes you've covered things many times, and sometimes you cover regions none at all. So if you want a whole genome, you can't just sequence 1.5 billion base pairs and get it. Actually, you need a tremendous amount of redundancy when you are only dealing with very small reads. Okay, so in this case, when you have long reads, you pretty much only need like 3x coverage and you're going to cover everything. And for short reads, for the same, if you sort of do a similar type of simulation or expectation, you would need roughly 8x sequence, which means 8 times the total length of the genome to guarantee that you're going to cover everything at least once. Okay, so that's one problem. That's actually not the big problem. Probably the big problem deals with the fact that when you look at a genome, it consists of regions that are single copy, which means they contain DNA that's essentially unique, and it also contains a tremendous amount of repeats. So here's a simple example. Let's say all the shaded boxes here consist of a simple microsatellite repeat, AT. Okay, so here's what happens when you sequence only these little short reads. So if you get a read that looks like AT, you don't know where it goes. You don't know if it goes here, you don't know if it goes there, or way over there. And actually, so the reads that we get generally tend to be 200 base pair segments on the end of a fragment. So they're what are called paired end reads. So you actually have a fragment of DNA that might be, let's say, several hundred base pairs long or several kilobases long. And the technology makes it easy to sequence a little bit on each side. So you can think of, you might have 2 pairs of sequences, a pair of sequences that you know are near each other, but if both of them show up as this AT, you don't know whether they map to these 2 or to other regions farther away. I mean, so it could be, this one and this one, or this one and that one. It's hard to tell. And the way that we try to figure this out is if you can sequence much longer fragments and you sequence their ends, if you can sequence sort of the edge here, where you know, okay, you've got some repeat and some sort of unique sequence, and the edge here where you have some unique sequence in the repeat, then since you know that this is actually, you know, a single fragment, that tells you, okay, there's a lot of repeats in the center, but it is a way of joining together, sort of mapping together this stretch of DNA with that over there. And so this is the basic conceptual idea surrounding how sequencing works now. It's you sequence fragments of different lengths, and you have these paired end sequences, and then you there are lots of pretty complicated algorithms for how you take all that data and try and come up with an assembly from it. Okay, so this is all just background to say you kind of need to have some kind of genome because you need to know where things are. And the sort of modern technology makes it so that you can generate this. This was done for, I'm trying to think of how much this costs. Probably on the order of about $15,000 in sequencing costs. To produce a total of roughly 70x coverage of the whole genome. And with these different sequencing, we sequence different fragments that range in average length of 350 base pairs all the way up to 10 kilobases. And when you do all together, you get a genome. One way of measuring the quality of a genome is something called the N50 scaffold size. So what that would be is half of your genome can be put into scaffolds, which are sort of like, you know, what we think of as sequence with gaps in it, that are 3 megabases or bigger. So you can think, you know, at least half your genome are in these fragments that are 3 megabases or bigger. Okay, so a genome is only sort of a first step. You know, if you're interested in admixture, then you need to do sort of a wider sampling. And so I'll just sort of give you an example of one thing that we've done. We took a single eastern barn owl that we think is not admixed, and we sequenced that to 6x coverage, and then we took another group of 16 spotted and barred owls sequenced at lower coverage. In this case, roughly half to 1x coverage for each of those individuals. And so here's sort of a preliminary analysis that we did. We identified particular nucleotide sites in the genome that are consistent with being fixed differences between spotted and barred owls. There are different ways you can do this. Here's one simple example. You could take sites that where this one barred owl individual, so that's the one that we sequenced at 6s coverage, we look for sites where they have 4 reads that support an alternative allele and 0 reads that support the spotted owl allele. It's not guaranteed to be a fixed difference, but it's highly enriched for them. And so we can do this, and there's millions of sites throughout the genome that satisfy that. And then we could just tabulate in all individuals the proportion of reads from the low coverage individuals that support what we think of as the likely spotted owl allele versus the likely barred owl allele. And so the basic thought is, if you have an admixed individual, okay, then that admixed individual will have some intermediate number of these reads that are supporting spotted owl versus barred owl alleles if their actual genome consists of some spotted owl DNA and some barred owl DNA. Okay, so that's the thought.
How did you assess the spotted owl allele if you didn't have a good genome of spotted owl?
Well, we have 170x genome. It's not.
Oh, I see.
So we can assess genotypes in that one individual, but we can't actually tie those. We can't always tie, you know, this stretch of sequence together with that other stretch of sequence or what chromosome it's on. But you know, we can identify orthologs very easily. Okay, so I should have made a figure, but I didn't. You can just look at these numbers. This is what happens. We're looking at the proportion of spotted owl alleles in a bunch of barred owl samples. Some of these are sampled from the west, some of these are sampled from the east. These are all spotted owl samples. The only thing to look at is all of these numbers are very similar to each other. Okay? All of these numbers here are very similar to each other. If in fact our sample included some admixed individuals, like if these western barred owls were actually admixed, you would expect them to look different than the eastern barred owls in this particular way, and in fact they don't. So they're, you know, these are all essentially identical. We don't find any individual that's sort of clearly showing some evidence of admixture. And if you sort of do some rough calculations, it's extremely unlikely that there's been more than 2% admixture in any of these samples. Now, we could probably do a little better analysis than that, but at least as a rough order of magnitude, there's not, you know, there's not an F1 in here. There's not a back cross. There's not even, you know, 2 generations beyond a back cross. If there's any admixture in those particular samples, it's very, very little. At least preliminarily, no evidence for recent admixture in these samples, but we still have phenotypic differences. So, you know, that slide I showed earlier with the, you know, the western barred owl in the center, there's something going on whereby to a trained curator's eye, it looks like an admixed individual in terms of having intermediate feather phenotypes and intermediate other, you know, gross morphology. So we don't really know what's going on. You know, either there is admixture happening and we haven't yet found it, we need to gather more data, or those phenotypic differences are maybe an example of convergent evolution. So there might be adaptation for being darker in color, for example, or being or having a smaller body size for the type of habitat. So this is sort of an ongoing thing that, you know, whether it's admixture or adaptation, in theory, if you gather enough genetic data, you'll figure it out. We thought it might be admixture, but it's not, obviously.
So Jeff, do your these samples include some that are phenotypically intermediate?
Yes.
They do.
Yes, it does include, in fact, that.
It includes the one you showed us.
Yeah, so it includes, Zach might know, I think it includes 4 or 5 individuals from a population that look exactly like this one here. So there's at least several individuals that were very likely, I mean, that should have been admixed if this were due to admixture. Sorry. Okay, so the last thing I'll talk about briefly, another project that I've been involved with that's looking at admixture between olive and yellow baboons. Okay. So, some basic background. Baboons are very widespread. They're very much not endangered. They're considered human. They're in some places they're human commensals and they're thought of as pests. Very smart and very strong pests, that is. And there are several distinct groups of baboons that differ phenotypically, morphologically, behaviorally, and they're sometimes classified as species or subspecies. It doesn't really matter which one. But they differ genetically much more than, let's say, different human populations do. So there's sort of a greater differentiation. And it turns out baboons are widely used in biomedical research. That is only of slight importance, but it will come back. So my collaborator, Jenny Tung, works at the Abicelli field site. And Abicelli is right here in this circle area here, and where they are, there are yellow baboon troops, but there's a hybrid zone that's very nearby, and their particular study site has been studied continuously since 1971, and continuously really means every single day, which is amazing, I think, and they identified something that it's starting in the 1980s. It looked like olive baboons, whose range is mostly up here, started expanding down, and so they sort of found their first documented male olive baboon immigrant in 1982. This is 30 years later. You can think of this is a crude genetic study where green is olive ancestry and yellow is yellow ancestry, and you can see there's sort of this pretty sharp gradient, and Amostelli is right there, mostly yellow baboon individuals and some recent migrants and hybrids and backcrosses. Okay, so here's where the fact that baboons are used in biomedical research comes in. So a draft genome of an olive baboon has been publicly available since 2008, but according to sort of the arcane rules of the Fort Lauderdale Protocol, if you know that, it means that even though it's public, you can't actually use it in your own work and publish on it, because they want to give the genome centers time to generate a publication. As it originally was formulated, that there is sort of an embargo time that was thought to be like on the order of 6 months to a year, but as it's actually interpreted, the embargo period is indefinite. So, as I said, it's been available since 2008. You can subtract as well as I can. It's now 2014, it's been 6 years. And the other thing is the quality of that baboon genome is actually pretty poor compared with other mammalian genomes. So, after a lot of trying to figure out another way around, we eventually decided to generate our own baboon assembly. And we chose a yellow baboon. It's still ongoing, our work, but I'll just point out a couple things. Our cost is a little bit less. I'm happy to make it available to anyone who wants it now, as opposed to never. And by at least this one standard measure of sort of how connected your assembly is, we can already do a better job than the public genome. So we already think we have a better genome, and we're still making slight improvements to it. Okay. So anyway, that's my little preaching moment. So we generated some low coverage sequence data from 10 olive baboons, 10 yellow baboons, and several baboons of known admixed ancestry from Amostelli. And with this slightly higher coverage here, we took a slightly more sophisticated way of trying to estimate local ancestry that essentially involves estimating allele frequencies in the ancestral populations, not sort of taking into account the uncertainty you have with low coverage sequence data. So you can, if you only have 1 or 2 or 3 reads covering a certain location, you can't really tell the difference usually between heterozygotes and homozygotes. And so you have to often work with what are called genotype likelihoods, the probability that you have each of the different genotypes. And so you can work with those directly and use that in sort of a likelihood-based approach to estimate sort of local ancestry, or local olive versus yellow baboon ancestry across the genome. So we did this. So first off, with 2x data is actually way more than enough in that if we run simulations, we should be able to pretty much exactly recover sort of the actual yellow ancestry, with our estimations. So the points here lie on the straight line. That's good. We can also confirm that we take individuals where we actually do know their ancestry, because of direct observation. And here we have F1s and backcrosses, and we can estimate so here's their known ancestry and that's their estimated ancestry, and we did a pretty good job there as well. So, so finally, so this is sort of ongoing. The one thing that turns out to be pretty interesting and worth following up on is that when we actually estimate ancestry across the genomes of the Amicelli individuals, we find actually lots of these really, really short tracts of ancestry. So little bits of olive ancestry in a larger sea of yellow baboon ancestry that suggests that this isn't the first time that there's been admixture of yellow and olive baboons. And so what we do know is that there is a hybrid zone, but the hybrid zone is currently moving, and it's potentially somewhere between possible and extremely likely that that hybrid zone has just moved one way and moved another way and moved one way back and forth as things like climate change, for example. And if you sample in a certain location, a fixed location, what happens is that population may be subject to periodic bouts of admixture. And this is what we think is happening. So that even though there's been a tremendous amount of information over the last 5 or 6 generations, corresponding to direct observations since 1970, we don't know exactly what happened before then, but the genetics suggests that maybe a few hundred years ago or sometime much further back in the past, there was also another bout of admixture, and this is something that we're going to pursue further. Anyway, I think I've gone over. So this is work that I've done with lots of other people, and I'm happy to answer any questions.